Hello students, welcome to the academia. Today our topic is cell division. Now the cell division is a continuous process and due to this the somatic cell and the germ cells divide. Cell division, before going to cell division we should know what about the diploid and haploid cell. So moving to that, diploid means what? Diploid is twice in. So a cell with 2 chromosome set in each of its cells are called diploid cells. And what about haploid cells? A cell with 1 chromosome set in each of its cells. So this is about the diploid and haploid cells. So generally our gametes like the sperm and eggs having the haploid kind of cells. And all the other cells of the body, that is all body cells, are the examples of diploid cell. Okay. So generally we can say that there are 2 kinds of cell in our body. One is somatic cell and germ cell. So before going to the cell division, the types of cell division, basically we focus on mitosis process. We should know about these preliminary things, that somatic cells, germ cells, haploid cells, diploid cells, etc. Next we move to the cell cycle. Now very simply, now we see the cell cycle. Now, the cell cycle consists of these following phases, and here we found this is the interphase stage. Interphase starts from G1, that is gap phase 1, S phase, and G2, gap phase 2. And then the divisional phase, this is mitosis, and mitosis is divided in prophase, metaphase, anaphase, and telophase. So in the mitosis phase the cell actually divide, and in the interphase stages there are these kind of divisions. Now in the synthetic phase there is synthesis of DNA, protein, and all the other things which is necessary for the division, and when from one mother cell to daughter cell form, before that there should be the doubling of all the contents of the cell so that the equal distribution occur in 2 daughter cells. The longest phase in the cell cycle is the interphase, and we can see that it consists of all these things. So this is very important. All living cells come from other living cells. It's true. During mitosis, the nucleus of the cell divide forming two nuclei with identical genetic information. Now we move to the mitosis process. So here comes the definition of the cell cycle. What is interphase? The interphase is the resting phase. In this phase, the cell replicates its DNA. It also produces additional organelles and cytosolic component, as I have already told, because from the mother cell there are two daughter cells and equal amount of content should go in two daughter cells. So in the interphase, there are high metabolic activity, but cell does not grow. Interphase consists the three phases which I have already shown you, G1, S, G2. So in the next diagram, you can see the actions or the functions which takes place in the three phases G1, S and G2. Now moving to the mitosis. Mitosis is the nuclear division. The process results in the exact partitioning of the genetic information. So the information which is present in the mother cell will equally distributed in the two daughter cells. In the mitosis, there are four stages, that is prophase, metaphase, anaphase and telophase. So one by one we will discuss the processes and in very brief way, not in such elaborate way, we just go to the brief manner all the four processes and what happens to these processes. So I will show you some diagrams and side by side the characteristics of these particular processes. In the process of prophase, what happens? So moving to the prophase, so in the diagram we can see that during early prophase the chromatin fibers condense. Generally when I discuss about the DNA, the chromosome, I have already told that the chromatin fibers are basically present and which condense to form the chromosome. In the early prophase, these chromatin fibers condense and their height shorten and they form chromosomes. The condensation process may prevent entangling of the long DNA strands as they move during mitosis process. So we can easily see that how the chromatin fibers condense to form the chromosome, and here in the process of prophase gradually in the late prophase there are nuclear envelope is a bit destroyed, the nuclear envelope, the nuclear membrane destroyed, and the fragments of nuclear envelope we can find. Next, the process of metaphase. So in the metaphase, the diagram shows that in this process the microtubules of the mitotic spindle align the centromeres of the chromatid pairs at the exact center of the mitotic spindle. And how the spindle form? The spindles are actually formed from the centrosome which divide and they form the spindle. And in the spindle, the chromosomes arrange and they form the metaphase plate. Here in the diagram, we can see the chromosomes. And they prepare themselves for the next phase. Next is the anaphase, and in the anaphase, the centromeres split, separating the two members of each chromatid pair, which move toward opposite poles of the cell. And this movement is called anaphase movement. Once separated, the chromatids are turned chromosomes. As the chromosomes are pulled by the microtubules of the mitotic spindle during anaphase, they appear V-shaped. So in the diagram you can see that what is the condition in the early and late anaphase stage. Next, moving to the telophase, the final stage of mitosis. It begins after chromosomal movement stops. The identical set of chromosomes now at opposite pole of the cell. In the diagram we can see this, uncoilate again and revert to the thread-like chromatin form. A nuclear envelope again forms, which was dissolved in the prophase. Around each chromatin mass, nucleoli reappear in the identical nuclei and the mitotic spindle breaks up. And we can see the cleavage furrow. The cleavage furrow occurred and it gradually divided the two cells, and it is called the cytoplasmic division or cytokinesis. So after the nuclear division, the cytoplasmic division occurred, and as I have told that by the formation of cleavage furrow, there is also division of cytoplasm. So in this way, in the mitosis process, from one mother cell, the formation of two daughter cells occur. So this is in brief, a very brief description of the 4 stages of the mitosis. Hope you people can understand this introductory part. If you have any query or question, you can ask or can comment in the comment box. Thank you so much.